gracias por venir a mi conferencia. Good morning, everybody. So today I'm going to discuss, so yesterday, if you were here yesterday, I discussed mainly some very general things about fluid dynamics and microfluidics. Today, I'm going to talk specifically about some work that I've done. Okay. So first, let's just do a brief review in case you weren't here yesterday or if you want a refresher. So yesterday I talked about a concept called electro-osmotic flow. Okay? And this is a way of moving fluids through very small tubes, very small passageways. Think about the size of maybe your hair or a little bit smaller. It only works in these cases. So remember that in general, a solid surface, when it's put in contact with water, becomes charged. Okay. As a result, the surface attracts some ions, right, from the solution that come very close to the charged surface and form what's called an electrical double layer. Okay. Now, when I apply an electric field along the channel, the ions move, and in doing so, they act kind of like a conveyor belt. Okay. They act like a conveyor belt that sort of drags the rest of the fluid into motion. Is there a laser pointer? Okay, that's it. So essentially, if you were to plot the velocity of the fluid versus position, you would see something like this. Okay. And remember that this layer of ions is extremely thin. Okay. If this channel right here were magnified to the size of this room, then that layer of charges would be about the thickness of the paint on the walls. Okay. So macroscopically, it looks very much like the flow is uniform. Okay, and then I showed this equation here. It's called the Helmholtz-Smalakowski equation. And it just says that the velocity, this velocity here, is simply related to the dielectric constant, the permittivity of the fluid, the electric field strength, the zeta potential, which remember essentially says how strongly charged the wall is, and divided by the viscosity. Okay? Remember that a very similar effect can happen if you have a suspended particle in solution. So if I have some kind of spherical particle that has say a positive charge, okay, and then I apply an electric field, then it will also move, and this process is called electrophoresis. Okay? Electrophoresis obeys a very similar equation to electroosmosis, and once again, it just depends on the charge in the particle and the properties of the fluid and the electric field. Okay? Great. So today I'm going to talk about some applications of this. Oh, great. Thank you very much. Okay. So for the first part of my talk, I'm going to talk about some work that I did for my doctorate, okay, my PhD. This was done at Arizona State University and the University of Washington. I want to briefly acknowledge my collaborators. So this here is Jonathan Posner, who was my PhD advisor, professor at the University of Washington. And then this guy over here is Philip Wheat, who's a classmate of myself and Professor Preguntas. And he and I collaborated on some of the experiments that I'm going to show. And he's now a fighter pilot in the U.S. Marine Corps. Okay, so let's imagine a situation. You have some rods, okay? We have some very tiny metallic rods that are made of half platinum and half gold. And when I say small, I mean specifically that these rods are 2 microns long, okay? So that's about 2 1,000ths of a millimeter. One way to think about it is if you laid 50 of these rods end to end, 50 of them, then you would reach the thickness of 1 human hair. Okay? I've held a small container in my hand before that has 100 billion rods in it. So these are very, very small. So why should we care? So here what you're seeing is simply the rods in pure water. There's nothing that interesting happening here. They're just vibrating and dancing around a little bit because of a process called Brownian motion. The way to think about that is they're colliding with water molecules. Okay? And so then, because of these random collisions with water molecules, they move around. Right? This process is very well understood. But now, let's see what happens when I add hydrogen peroxide to the solution. Okay, so here we have about 5% peroxide, and we see this motion, this swimming motion, in addition to the random Brownian motion. Okay? Now, you can't really see from this image, from this video, but one interesting thing is that whenever they swim, the platinum end is always forward. Okay? Platinum end is always facing forward, and the gold end is facing backward. So that would seem to give us a hint as to how this works. But the basic question is, what is going on here? And this was the original question that inspired me to begin my work. So, as you may know, this sort of thing happens all the time in biology. I talked yesterday about swimming at the micro scale and how it's difficult, right? It's kind of like swimming in caramel, you know? It's like if one of us tried to swim in a very viscous fluid. But there are countless types of organisms in biology that can actually accomplish this, and we should be grateful for that, because all of us are alive thanks to the ability of sperm cells to swim through the mucus in the cervix to actually fertilize an egg and begin the process of pregnancy. Okay? There are many other bacteria, such as E. coli, right here, that can swim all the time to essentially locate nutrients to find food. Chlamydomonas, right here, is a species of algae that's found in the ocean, and it swims the breaststroke. Paramecium here is a slightly larger organism that swims using these little hairs here. They're called cilia, and they use these cilia to avoid predators. Okay, but what you may notice here is all of these organisms swim mechanically. They have some kind of appendage, like a hair or a flagellum, and they wave it around, and then they move as a result of that motion. Okay? But these rods, okay, these rods don't have any moving parts. They're just little pieces of metal. So how could they possibly be propelling themselves? Right? So it has been proposed before that some bacteria might be able to swim without waving their flagella. Okay, and the mechanism here was proposed by a scientist named Peter Mitchell, the British biochemist, and he won the Nobel Prize in 1978 for some different work that he did. But in 1956, he gave a talk where he suggested that some bacteria could possibly move by some mechanism like what you see here. Okay. And so he said the mechanism I'm suggesting amounts to the use of a stream of ions passing within the organism in one direction. Okay, so imagine that you have ions conducting through the tail here, and over its surface in the other. Okay, so then outside of the organism there are some ions going backward. Much as a caterpillar track is used for the locomotion of a tank. So by caterpillar track we mean one of these things, right? So if this tank wants to move forward, the track has to move backward. So he was essentially proposing that a similar sort of phenomenon could be happening with these bacteria. So the important thing about this, though, is that he was only speculating. He wasn't actually claiming that this was reality. He was just proposing this as an idea. Meanwhile, a few years later, so if anyone's wondering where I got my hairstyle from, this compare here. He pulls it off a little better than I do. But so Richard Feynman, of course, was a very famous physicist, and he gave a very famous lecture in 1959 called "There's Plenty of Room at the Bottom." This talk has been considered as the foundation of the field of nanotechnology. Okay? And toward the end of his talk, he proposed making a tiny electric motor, a tiny machine that could do some kind of useful task that is smaller than 1/64th of an inch on one side. Now, his challenge was soon satisfied, so an American engineer named William McClellan actually was able to make a tiny motor, but it wasn't very useful for anything. It just kind of spun around. Right, so we're still looking, even in 2015, we're still not sure if there's if we're going to be able to find a useful microscale machine. Now, so coming back to biology, in 1985, these people discovered Synechococcus, which is a bacterium that's found in the ocean. And these bacteria can swim, but you'll notice they don't have any flagella. They don't have any kind of appendages that they could wave. So the question is, how do they swim? Well, it was proposed that they might swim using a similar mechanism that Mitchell proposed. Okay? That maybe they're able somehow to generate an electric field, okay, that will then cause ions to move along the surface here and propel the organism kind of like the caterpillar track. So Anderson proposed that this formula might apply to those bacteria. So I just want to point out this grouping of terms right here. This should look familiar from yesterday. So again, we have the permittivity, zeta potential, and the viscosity. This eta here is viscosity. And this is called the electrophoretic mobility, these grouping of terms. And then here, this grouping of terms is essentially a self-generated electric field. So they call this self-electrophoresis, right? Because the organism is moving by electrophoresis, the same animation I showed you, but instead here, the electric field is generated by the organism itself. However, in a few years later, Pitta and Berg conducted, what they did was they applied an electric field to a suspension of the swimming bacteria. Okay? And remember that for electrophoresis to occur, or self-electrophoresis, you need a surface charge. The bacteria needs to be charged. Well, what they noticed was they measured the electrophoretic mobility, essentially the zeta potential, and they found that it was about 0 for these bacteria. Right, so what that says is that self-electrophoresis is not the mechanism, because if they don't move in an electric field, then there's no way they can propel themselves by electrophoresis. And so even today, we still don't really know. We have some ideas. These guys proposed a new mechanism, but it's still not confirmed. We still don't know exactly how these work. So later, the next year, we have a new study that looks at another theoretical study of a spherical cell, and essentially they were just analyzing the case where the surface charge on the cell varies from front to back. So at the front of the cell, the surface charge is positive, okay? And at the back, the surface charge is negative. So that will generate these electric field lines, okay, that start here and end here. And if the cell has a zeta potential, then it should move according to this velocity. Okay? So this is just another theoretical proposal for this mechanism. So just a brief, I've shown you a lot of history, so I just want to summarize it. So for about 50 years, we've been thinking about possible ways in which a small organism might propel itself by generating its own electric field and then moving in response to that electric field. But we have never seen an actual example of it until now. So I want to talk briefly about some man-made swimming devices that have been made. So recently these researchers at University of California demonstrated that they can make small swimming particles that can actually deliver drugs in the stomach of a mouse. Okay, so this is kind of the first step towards actually using these swimmers for something useful. Okay, I haven't shown you yet that these swimmers that can carry cargo. Okay, and several other propulsion mechanisms have been investigated as well. So I'll just briefly show them. There's self-diffusiophoresis, self-electrophoresis. There are a couple of different types of a couple of different realizations of that. And then we have thermophoresis, in which a particle generates a gradient in temperature and moves in response to that. But today, I want to focus on the platinum gold rods that I showed you earlier. Okay, so again, our interest is in these bimetallic nanorods. They're about 2 microns long, about 300 nanometers in diameter, and they swim in hydrogen peroxide, always with the platinum end full. So the more peroxide you add, the faster they swim. If there's no peroxide at all, as you saw, they don't move at all. So the peroxide is like a fuel. Now, other metals can also be possible, so you can make a palladium gold rod, platinum palladium, etc. And we can actually predict the motion in each case. We can predict the direction it will move. So what are these useful for? Well, one thing we can do is pick up and release cargo. Okay? So what you're seeing here is a little swimming rod, and we have a magnet actually to guide the direction of motion. And we're able to control where it picks up the cargo. So the cargo also has a magnetic coating on it, so the attachment is by magnetism. And so it picks up the cargo. We can control where the cargo goes. And then notice right here, we spin the magnet very quickly, and the rod essentially breaks off of the cargo. So imagine if you ever play with 2 magnets, right? If you twist one very, very quickly, you can separate them. Okay, so this is a proof of concept experiment. And so we can imagine, well, what if this was a cancer drug? Okay. Well, then potentially, maybe in a couple of decades, we could see these rods used to deliver cancer drugs in the body, okay? Because we can control where they go. So that's still an application that's under exploration, but it's a possibility. Okay, so, but what I'm interested in, see, we won't really know exactly what we can use these rods for until we know exactly how they work. Okay? So my questions, the questions of interest, but first, well, how does this even happen? How do the reactions cause the propulsion of the rods? How does the speed depend on parameters of the system? How does the speed depend on the concentration of hydrogen peroxide, the temperature, maybe the viscosity? We don't really know. And finally, what are the limitations on the rod's capabilities? What can they actually do that would be useful for humanity? So to answer this question, I constructed a mathematical model of the rod's motion, and we conduct simulations of the rod's motion to try to understand the physics. Okay? So I'll just briefly go through the equations. So this equation here is called the Nernst-Planck equation. It describes the concentrations of different species in the solution. Poisson's equation describes the electric potential field. Okay? So this is how we're going to describe the electric field in the solution. And the Stokes equation, well, this Stokes should be familiar to you, because I showed yesterday the Navier-Stokes equations. The Stokes equations are a specific simplification of the Navier-Stokes equations for a very low Reynolds number situation, right? Remember the Reynolds number? The relationship between inertia and viscosity. So we're very, very small. We're in a very, very small size regime here. So this is where the Stokes equation would be appropriate. And finally, we can, just based on the governing equations, we can predict how the speed will depend on these parameters. So what you're seeing here is the electrophoretic mobility, again, dielectric constant, zeta potential, and viscosity. J is the reaction rate. So what you see here is that the rod is essentially acting like a battery or a fuel cell. Okay? So on platinum we have oxidation of peroxide. So we take peroxide and we produce protons, oxygen, and electrons. Those electrons move through the rod, right? It's conductive. The rod conducts electricity. And essentially protons are released into the solution all around here. On the gold side, peroxide reacts with protons and electrons and produces water. Okay? So J, right here, J is the reaction rate. It's the amount of protons that leave the surface per unit time. Okay? And so it makes sense if we increase the concentration of hydrogen peroxide, for example, the reaction rate should increase and the rod should go faster. And finally here we have the ionic strength, essentially the total concentration of ions, or electrolytes, in the solution. So this equation predicts that the speed is inversely proportional to the ionic strength. And that will be important later. So how do we describe what's happening at the surface? Okay. So this diagram hopefully looks familiar. And the basic idea here is that there's a layer of charged ions and water molecules that essentially sticks to the rod. Okay. And so that's called the Stern layer, and it has a thickness of lambda s, only a couple of molecules. And as a result, to describe the boundary, the electric potential distribution, we need this mixed boundary condition. Okay, so zeta, the zeta potential is the potential evaluated at the outer edge of the Stern layer, and it decays out to the bulk value of 0. And then the Stern layer, we model that as a simple capacitor, and the potential essentially changes linearly across the Stern layer. And so there's a voltage across the Stern layer that we care about. Generally, the voltage actually turns out to affect the electrochemical reaction rates. But in general, here, the electrochemical reaction rates are described by these simpler expressions. We're essentially simplifying the equations a little bit. So I guess the basic idea here is that the rate of reactions on the platinum and the gold depend on the peroxide concentration, and in the case of gold, they depend on the proton concentration. Finally, we also have to make sure that the total charge in the rod does not change with time, right? Because we're essentially modeling the steady state behavior. So the rod is moving at a constant speed. Nothing is changing with time. So imagine essentially the amount of protons coming out of the platinum surface has to be equal and opposite to the number of protons coming into the gold surface. Right? If this is not true, then the amount of charge then that would imply that more charge is entering the rod than leaving the rod, or more charge is leaving the rod than entering the rod, in which case the amount of charge in the rod would be changing with time. So that's not allowed. Okay. So that's all the math. That's all the math I will show. Okay. So now let's show some results. What you're seeing here is a rod. So these are results of computer simulation. These are results for a rod in a peroxide, or just in water. And what you're seeing here is a negatively charged rod. And in color, you're seeing charge density. It's essentially how strongly charged the fluid is at a given point. Okay? So remember that the ions will form an electrical double layer that has positive charges in it, right? If the rod is negatively charged, then the charge in the electric double layer should be positive. And that's what we see. So if charge density is positive, that means the fluid is positively charged. So here, I have not included the reactions yet. So here we're only including the reactions and not the surface charge. So remember that the reaction on platinum generates protons, okay? So protons are positively charged. So when protons leave the platinum, then there should be a region of positive charge here. When protons are consumed on the gold side, then we should have a negative charge right here. So, and that's indeed what we see. So again, this is charge density. The important thing here, though, is that the fluid is positively charged near platinum and negatively charged near gold. As a result of the charge, then we have this electric field distribution, where we have electric field lines that point from the platinum to the gold. So, in reality, we have both of these situations. We have, in a real situation, we have a rod that is charged, and we have a rod that has reactions happening on it. So now let's combine the two. So if we add these two distributions together, we see this. We see another distribution of charge density where the fluid around platinum is primarily positive. So now consider this. So remember back to the electro-osmosis or electrophoresis diagram, right? So I have an electric field that points along the surface. Okay. As a result, the ions in that layer will move along the surface, and they drag the fluid with them. Well, we have the same thing here, where we have some positive charges in the rod. And notice that this electric field, well, it has this kind of complicated shape, but there are components, some of the electric field is pointing from platinum to gold. And so we should see an electro-osmotic flow from platinum to gold. Okay. So again, think about squeezing an ice cube, right? When you're squeezing an ice cube, you kind of apply, with your fingers, you apply a force that's that way, and then the ice cube goes this way. So what you're seeing here, we should see fluid moving from front to back, and the rod should move forward. And that's indeed what we see. So here, you're seeing, in color, I'm showing the fluid speed. And this is from the perspective of the rod. So imagine you're sitting on the rod, and you're watching the fluid come past you. Well, then, the fluid out here should be moving backwards, and you are moving forward. So, again, the motion of the rod is this way, with respect to a laboratory reference term. Okay, so the punchline is, with these simulations, we can predict how fast the rod will be moving for a given situation. And so we can plot this. So in blue, I'm showing the speed of the rod according to the simulations. And in black, I'm showing the speed of the rod according to experiments. Okay? And there's pretty good agreement right here. There's a little bit of a difference right here, and there are several possible reasons for that. The main reason, I think, is that these rate constants here tend to vary with the peroxide concentration. We're not including that here just for simplicity, but these rate constants actually it's very possible that they could decrease out here and increase here. And if that were to be the case, then the agreement would be improved. But the agreement still is quite good. So now I just want to briefly show, remember I said that the speed should depend inversely on the ionic strength or the conductivity of the solution. What that means is, if you add salt to the rods, to the swimming rods, if you add salt, that will actually slow them down. And that's been observed experimentally. So in black here, I'm showing measurements of Paxton and co-workers, Walter Paxton, good friend, and they showed that the speed slows down as you increase the electrical conductivity of the solution. Now with the simulations, we're able to predict that speed as well. We can actually show that in the simulations that the rods also slow down. So again, we see fairly good agreement between experiments and simulations. But we also see good agreement with a formula, an analytical formula, provided by Golestani and co-workers. And that formula is shown right here. So L is the length of the rod, D is the diameter of the rod, mu E is the electrophoretic mobility I showed earlier, epsilon times zeta divided by viscosity. I is the electrical current coming out of the rod, essentially the flux. the reaction rate, and sigma is the conductivity. Okay. And so we see excellent agreement between my simulations and Golestani's formula at high conductivities. The agreement starts to deviate a little bit at low conductivities, and we think we understand why that is. There are certain assumptions that Golestani made that affect the accuracy of his prediction for low conductivities. Okay, so again, essentially the model allows us to predict how the motion will depend on system parameters. So I just want to give a brief summary of what we've learned about this from this model. Okay, so first, the electrochemical reactions between peroxide and the rod that occur on the surface create this asymmetric charge distribution, right? Remember you saw that the fluid is positively charged near platinum and negatively charged near gold. Okay, that essentially creates an electric dipole, but then creates an electric field that points from platinum to gold. Then that electric field then acts, it drives motion of the particle. So in conclusion, this is really the first realization, I think, of the mechanism that Mitchell proposed over 50 years ago. So there are many unsolved problems with this work. So I mentioned that if you add salt to the solution, it slows the rods down. That's true, except if the salt contains silver ions. Okay. So it was shown in 2009 that if you add a salt that contains any one of these ions, it will slow the rods down. The speed goes down. Except if you add silver ions, it speeds them up. And again, this is not well understood. Not well understood exactly why this happens. There are some hypotheses about deposition, saying that maybe the silver ions can deposit themselves onto the gold surface and somehow increase the reaction rate and increase the speed. But again, that has not been investigated in detail. So there's plenty more room, I think, for investigation here. Okay, so with that, that's the end of the first part of my talk. I want to pause here and see if there are any questions so far. I've been talking for a while. All good? Okay, great. Okay, so that was my PhD work. I completed that in 2013. And now I want to talk about some work I've done at MIT. Yep. Oh, question? Yeah. Yes. Yes. Energy harvesting. Okay, so harvesting energy from the rods. Yes. It's not something we have looked at in particular. So you're saying maybe we could harness the energy from the rods to accomplish some kind of useful task? Probably. And also sorry. Yeah, to use them for something useful. Yeah. As far as energy harvesting goes, I think that would be difficult just because these rods are so small that the amount of energy each rod has is very tiny. Right? These rods are only about 2 microns in length. The main application that we are considering for this would be for chemical sensing. Okay, so chemical sensing. So imagine if you have a sample of drinking water. If you have a sample of drinking water and you want to know if there's a silver impurity in the drinking water, maybe you're concerned about too much silver. Okay, well then you could drop some of these rods in here, and you add a little bit of peroxide, and if the rods speed up, then you know that there's silver present. But if they don't speed up, then you know that silver is not there. So they couldn't. That's kind of the most straightforward application I can think of. Maybe somehow, if you're able to get a lot of these rods to swim together, then maybe we could talk about doing some kind of energy harvesting, because then that would be more, there would be more energy there to get. But then that would still be difficult because in order to make the rods move in one direction, you have to use a magnet. You have to have magnetic guidance. You actually have to supply some energy to the system. So without any kind of guidance, the rods kind of swim in. True. So they could possibly, right, in regions where, yeah, so maybe look at from the Earth's magnetic field, or, yeah. Well, the Earth's magnetic field by itself would not be significant, I think, enough to, at least under normal laboratory conditions. Maybe if you found a certain area where the magnetic field was very strong, then that could potentially work. Yeah. It's a good question. It's not something we've really thought about too much, but those are kind of my initial thoughts.
Okay. Also, I'd like to find some connection with the way the organisms, human beings, manage the energy at those micro levels.
Right. So how like bacteria manage their energy.
Not also bacteria, but also the food itself. Because you produce hydrogen.
Yeah, carbohydrates, yeah.
And very wisely the organism manages to convert.
Yeah.
By this kind of processes also.
Right.
Quick processes between lines and
yeah.
that.
Right. Yes, and actually many organisms use swimming. They swim to find food. They find nutrients here. And rods can do something. The rods actually do the opposite though. The rods actually, if you put them in a gradient, where a channel where there's lots of peroxide on one side and not so much peroxide on the other side, and if there's lots of salt on this side and not very much salt on this side, over time the rods will actually drift and they'll all aggregate over here. Right? So they actually swim away from their fuel. So it's kind of counterintuitive. But so I'm not sure you would necessarily see the same behaviors with these rods that you would see with, say, E. coli, right? Because E. coli can actually sense regions where there's not so much food and regions where there's a lot of food, and they can turn around and actually find the regions with lots of food. So that it's a little bit, it's kind of the opposite effect. So, does that help? Any other questions? Okay. Great. So here, so now I'm going to transition to my postdoctoral work. So I have many collaborators here. The principal investigator of the group is Professor Colin Dewey. I also work with a graduate student in the group named Jifei He. He's from China. And a good friend of mine, Pablo Garcia, native of Cali, Colombia. And so I'd like to acknowledge all of them. Paulo's another postdoctoral researcher in the group. One of the great things about working at MIT is that you get to meet people from all over the world. It's completely normal in the course of one day to hear about 5 different languages being spoken, and it's really great to see such a diverse array of different perspectives. So first, I'd like to show. So I want to give you a basic idea. So here, we're working with a process that's called electroporation. Okay. So what is electroporation? Electroporation is a technique in which you have a cell, okay, and cells generally have lots of ions inside them, and it's in some kind of solution that also has lots of ions. Okay. And I position the cell between 2 electrodes, and I'm going to apply a very high voltage between those electrodes. Okay. So that creates a very strong electric field, and the charges, in response to the electric field, the charges inside of the cell will reposition themselves, and they create this kind of a distribution. So that, notice here we have lots of positive charges on the inside and lots of negative charges on the outside. You have the opposite over here. So that actually creates a voltage across the membrane. It's called the transmembrane voltage, okay? The TMV. And so we can predict how this transmembrane voltage will look for, at least in this sort of simple picture, using this equation here. So let's think about what's happening at the cell level on the membrane. So cell membranes, as you may know, cell membranes are made up of lipid bilayers. One of these little guys is a lipid molecule. Okay, and lipids contain hydrophilic heads and hydrophobic tails. So hydrophilic means it likes water, and hydrophobic means it does not like water. So essentially the lipid heads are in contact with the solution out here, which is water, and the inside of the membrane is in contact with the inside of the cell, which is mostly water. So as long as the transmembrane voltage is below 1 volt, then these lipids, they'll be constantly fluctuating because of thermal energy, and they create these small hydrophobic pores. Okay? So that's not too interesting. That's happening in every one of your cells right now. What's interesting is that when you add even more voltage, so that the membrane voltage across the membrane becomes greater than 1 volt, then these pores can actually begin to appear, these hydrophilic pores. So the lipids actually reorganize themselves, and they form these 5 nanometer or so pores. So it turns out that those pores can allow materials like DNA or drugs to actually enter into the cell. We can use this technique to introduce material into the cell. Okay, but what is electroporation used for? What's it good for? So for that, I want to show briefly some of the work of my collaborator. So this is work that's done by Paulo, and so this is on a technique called irreversible electroporation. Okay? So the idea is this. With electroporation, if you apply a strong enough electric field, then you can actually kill cells, right? You can imagine if you just keep increasing the voltage further and further, you can actually damage the cell so badly that it cannot recover. And if it's a bad cell, like a cancer cell, then that's good. We want that to happen. That's called irreversible electroporation, IRE. And so irreversible electroporation is actually being used to treat cancer. Okay? So what you're seeing here is some work done. This is all work done by Paulo. And this was for his PhD thesis. So I'm just going to briefly discuss what he did here. So the idea is to treat a brain tumor. This is a scan of a brain tumor in a dog. And the tumor is right here. And so what Paulo did is he created a computational model, of the brain. And these are electrodes right here that are inserted into the tumor. And then what's going to happen is he's going to apply a very high voltage across these electrodes and generate this electric field distribution. So in color, you're seeing essentially the intensity of the electric field. Okay? So the point is here you can predict how the electric field will vary with position. So you can, right, because you can imagine you don't want to damage healthy tissue. Okay, so then the basic process for treatment planning here is as follows. So I'll just go briefly through the process. So first, you acquire medical images, say from MRIs, a CT scan, etc. Next, we perform segmentation of tissue. So, where's the tumor, where's the white matter, the gray matter of the brain, the ventricles, and more specifically, where are we going to insert the electrode so that we can kill the cancer cells but not healthy cells. Okay, then we create the computational model, determine the tissue properties, right? So we have to estimate the electrical conductivity of tissues. And then we do a parametric study, so determining the voltages that need to be applied, actually perform the treatment, and then finally refine and validate the models. Okay, so here, these are some more examples of the actual therapeutic planning. So here we're actually showing what happens, when the electric field is applied for different voltages. Okay. Okay, and these are actual results from an actual canine patient. So before irreversible electroporation, you'll see there's the tumor is located right here. 4 months after the irreversible electroporation treatment, the tumor is significantly reduced. And then 9 months after, it's essentially disappeared. You can just bear. The point is the tumor has been reduced in size because of the electroporation. Okay, so I just wanted to show a couple of videos. So this is an actual patient. And before the treatment, I think in this particular case, the cancer was located in the front legs. Let's slow it down this morning. Catheter crowns? Okay. So before the electroporation treatment, it's having lots of trouble. Just walking around. 72 hours after the electroporation treatment. Don't eat that baby. So this treatment is successful, okay, at least for this particular case. And irreversible electroporation is actually now being explored as a possible cancer treatment for humans, for several different types of cancers, pancreatic cancer and prostate cancer, just to name a couple. Okay. Sorry, 1 second. Very rapid review of what we've talked about. So now today I want to talk about a different application of electroporation. So our interest is in genetically engineering bacteria. Bacteria can be engineered to do lots of different things. One example would be cleaning up oil in an oil spill. So if you give a certain piece of DNA to a bacterium, it will actually acquire the ability to eat up the oil. You can also make bacteria that will act as catalysts in a microbial fuel cell. Okay, so this is one example of how that could work. The problem is, it's very difficult to use electroporation to insert DNA into bacteria. For most bacteria, we don't know the proper conditions. We don't know the voltage, we don't know what kind of solution we should use to actually successfully get the DNA into the bacteria. So electroporation protocols only exist for a tiny fraction of bacteria on Earth. There are about at least 11,000 different species of bacteria, but we only know how to do electroporation for about 100. So the goal of this work is really to understand the factors, the principles that control success. What controls success of a particular electroporation experiment? So again, the primary research question is, what are the conditions that control that essentially maximize the likelihood of success. What are the conditions that are most conducive to success? And when I say conditions, I mean the electric field strength, the electrical conductivity of the solution, the duration of the electric pulse, right? So we could have a longer or shorter pulse. We could have different types of pulse shapes. So we could have the voltage increase smoothly and then drop, or we could have a square wave, right? There are a lot of different parameters to explore. But the idea that we had, so Paolo and I were just talking one day, and we came up with this idea here. We said, what if we make a microfluidic device that has a channel that constricts, that converges and then diverges? Let's say that we fill that channel with a suspension of bacteria, and this dye here, this fluorescent dye is called Cytorex. So there's a dye that only lights up, that only gives off light if it's inside of a cell, okay? So the idea is that we perform electroporation inside this channel, so we apply between 1,500 and 2,500 volts over a period of 1 millisecond. And because the walls constrict, well, PDMS, or the material of the device, does not conduct electricity. And so what will happen is the electric field will actually vary with position. And only a certain subset of cells will undergo electroporation. The electric field where the position where we first see fluorescence is actually where the minimum electric field is for that particular cell. So briefly, I want to discuss how microfluidic devices are made. So we start by making a master through a process called photolithography. And so the basic idea is that you have a silicon wafer, and it has these blocks patterned onto it, okay? These blocks are the same shape as the channels that you eventually want to make. So then we pour essentially a polymer solution called PDMS, polydimethylsiloxane, and then we cure it. We cure it by baking in an oven. And what happens is that polymer will solidify, become solid. And it's kind of like, PDMS is kind of similar to like a contact lens, like the same kind of soft, squishy polymer material. Then we peel the PDMS off, and so now we have a stamp, right? And so these channels here can now be bonded to a piece of glass. Okay. Now we can actually flow fluid through these channels. And so to create this microfluidic device, we did exactly that process. Okay? So I mentioned that the electric field varies with position, right? So this is a COMSOL simulation. COMSOL is a COMSOL multiphysics. It's a modeling software. Okay, so we have electrodes right here and right here. Okay. And we apply the electroporation pulse, and as a result, we see this continuous variation in the electric field strength. So for several different applied voltages, you can see that the electric field varies linearly with position. Okay. And that's important because now, let's say that for a given bacterium, the bacterium needs to experience an electric field of 8 kilovolts per centimeter, okay, in order to experience electroporation. And so then you would only see, only in this region, you see cells that have taken up the dye, right? Because then only these cells undergo electroporation, so only those cells actually have holes in them that the dye can go through. So does this experiment work? I mean, everything I've proposed so far is just an idea. So here we have the actual microchannel. This is an actual microscope image. And there are a few cells that are already fluorescent, right? So it's possible that the dye can sometimes get into the cells without electroporation. but mostly the cell is dark, or the channel is dark. Not a lot of fluorescence. When we apply the pulse, we see fluorescence. Okay. So the experiment does work. Okay. But now our job is to determine where exactly the fluorescence increase because of electroporation. Where does that actually happen? Right? It's kind of difficult to locate exactly where that begins. What's the actual difference between these two sets of data? So we can do that a few different ways. So what I'm showing here is the fluorescence intensity integrated over a certain portion of the channel. So let's see. Yes. Okay. So imagine the idea is this. So we take essentially the fluorescence data here, and we add up the fluorescence in each one of these pixels. Okay. We keep adding and adding and adding, starting from the right. We keep adding it up, and we look at two different data sets. So we look at before in the red and after the pulse is applied. And so you'll notice they both, the two data sets start to diverge. They start to separate right at about a very definite place. Okay. So this is one way to sort of visualize the change in fluorescence for before and after, right? Because the after data, the blue data, should have a higher fluorescence, and so then when you integrate the fluorescence distribution, then the slope will be higher. So this is fine, but it's not a very rigorous method for determining the location. So I want to talk briefly about, so here, let's consider a simple case. So let's just plot the sum of the fluorescence in each one of these pixels, okay? And let's plot that on line. So here you're seeing just the fluorescence intensity before the pulse in red, and the intensity after the pulse in blue. So what you notice is, for certain parts of the channel, the fluorescence before and after the pulse is about the same. That's because in these regions, electroporation has not occurred. And so the dye is still outside of the cell, and it's not giving off any fluorescence. In the regions where the electric field is high enough for electroporation, then the dye actually goes into the cell, okay, and the fluorescence is increased. So using a statistical test called the KS test, the Kolmogorov-Smirnov test, we can actually distinguish between the before and after data. So very briefly, the Kolmogorov-Smirnov test is a very well-established statistical test to determine if two data sets are similar in some sense. So essentially, we implement this test in MATLAB, and we consider this null hypothesis. And if the test returns a value of 0, then that's saying that the test does not think, the test thinks that the two data sets are similar. So we should see a value of 0 right here, right, where the two data sets are similar to each other. When we see a value of 1, that indicates that the data sets are different. Okay. So we can do that, and we can actually determine the critical electric field values. Okay. So again, here, for this portion of the data, the statistical test is saying that the before and after data sets are similar, and so electroporation has not occurred. So basically, wherever H equals 1, that's a region where electroporation has occurred. Okay, so then we have, so this is some data for 3 different bacteria. So the critical electric field for 3 different bacteria, E. coli, Corynebacterium glutamatum, and Mycobacterium sphegmatis, are all plotted here. So each one of these bacteria has some important applications. E. coli is used in many biological studies of protein expression. So, for example, if you want to study how a certain bacterium takes up and expresses a certain protein under different conditions, then you would use this E. coli. Corynebacterium glutamicum is used to make a variety of different products, vitamins, amino acids, etc. And Mycobacterium phygmatis is used to study, as a model organism to study tuberculosis. And what we're seeing is that there's a fairly well-defined minimum electric field for each bacterium. Okay. So we believe that this assay is a very robust way to quickly determine the minimum electric field required to electroporate a certain bacteria. And that's important for designing protocols, for example, to genetically engineer bacteria in the future. So in conclusion, we invented a new method to determine the minimum electric field for electroporation. The key to the success of the method is that we can continuously sample a continuum of electric fields. So you can imagine if you wanted to try to determine the electric field, the minimum electric field for using other methods, if you only tried one electric field value at a time, you would have to do hundreds of experiments. But here we do all those experiments at once. The analysis uses very small amounts of sample, less than a microliter. And it could also be used to study transformation of mammalian cells, like human cells. Okay. So we believe this device would be very versatile for other situations. Finally, we can use this device, again, as I said, to design electroporation protocols for other cells. And specifically with bacteria that we were unable to treat with electroporation before. Okay, so that's about it. I wanted to ask if there are any questions about this portion. Yes, in the back.
Jeff, you was talking about treating cancer in tumors where it's focused. Yes, but what if it is not focused? Yes, like cancer in blood or in bones, you know.
Right, so that's a good question. And in general, it would be more difficult, yeah. In fact, I'm not sure that cancers of the blood would be treatable using this method. I think the point is you have to, for this particular treatment, the placement of the electrodes is very, very specific, yeah. So, as I mentioned, this is applicable to certain types of cancer, like cancer where there's a specific tumor. There's a tumor in a specific location. I think for cancers like leukemia, that would be much more difficult. Paolo might be able to tell you further information about that. It's possible that you could, if you locate a certain region, maybe where the cells are especially concentrated, or if you could isolate those cells and take them out of the, you could potentially get useful information if you could extract some of the cancer cells that are circulating in the blood, like circulating tumor cells. You could try to extract them and perform this electroporation test on them in vitro, outside of the body. And that could give you information about what it would take to kill those cells. But I think as far as an actual tumor treatment, this technique would not be applicable. So that's a good point. Other questions? Yes?
Have you thought on electroporation on neuronal processes where the flow of ions are so important?
Mm-hmm. I think, I have not looked at that specifically. So you're right that it would probably significantly disrupt the activity of the neuron, right? So if you were to treat, if you were to apply an electric shock across an electroporation pulse across let's say a neuron, that could significantly disrupt, say in the brain. Maybe you're thinking about what happens to healthy tissue in the brain near the tumor, right? The action potentials of that the neuron will generate would be significantly disrupted. And actually, that's one of the reasons that the modeling is so important. Is because you have to be able to place the electrodes in just the right place so that the tumor cells are killed, but the damage to the healthy brain tissue surrounding the electrodes, surrounding the tumor, is minimized. Yeah, I imagine there could be some of that could be happening to some extent. It's definitely it's minimal. It's minimal because Paulo did a lot of very extensive parameter. He looked at different voltages and looked at where what voltage is high enough to kill the tumor cells, but is not so high that the electric field in the healthy tissue is very high. Right. Okay. Other questions? Well, if there are no other questions, then I just want to thank you all for a really wonderful visit. I've had a great time, and I look forward to speaking with, talking with all of you in more detail about what you're doing, and if you have any questions about what I'm doing. So, thank you very much.
Universidad Tecnológica de Pereira.